Hello everyone, many thanks for joining this live speaker Q&A. It's my pleasure to welcome our speakers to this session. A quick note for attendees, please do comment any questions that you have via the platform on the chat feature there, and we'd be delighted to answer them. You're also welcome to join in the Zoom call and you can ask your question to the speaker directly. So thank you so much, Nathan, for joining me. I've got a question here for you straight away, which is, what other multivariate analytical techniques are useful for lymphocyte population-based analyses of clinical data sets?
I think the question that one is trying to address will dictate that. In my presentation, I used a quadratic discriminant analysis algorithm. There is a linear version of this called linear discriminant analysis. And these are supervised algorithms. In other words, I know a priori how I want to categorize the data. I knew that there were 4 treatment arms, and I wanted to see if the lymphocyte response or the immune response was distinct enough to categorize patients into 4. Many people take an unsupervised approach, and they used kind of a categorization algorithms like principal components analysis, which is looking at variance among the groups, and it's completely, you have no control. You just say, show me where everything clusters. There's another technology that I'm very fond of called Kohonen maps, or self-organizing maps. They're named for a Finnish mathematician named Tuvo Kohonen, who I believe is still a practicing mathematician. And these are very similar to principal component analysis, and they form a map. And all of these have in common, they put like populations closer together and dissimilar populations further apart. And then finally in flow cytometry, I think we're all aware of many of the algorithms like stochastic neighbor embedding or SNE or spanning density tree analysis or SPADE, and various other algorithms to, and they all have in common the one thing, and that is to cluster like observations together. But it's really a matter of the question one's trying to answer.
Fantastic. Thank you so much for that. You moderated the panel discussion earlier, which was excellent, and thank you so much for doing that. We did have some questions that I don't believe there was time to address. So the first question here is there a publication that you would recommend with respect to setting up robust analysis workflows?
I wish I could. Right now, there are multiple industry consortia that are focusing on that. How do we analyze all of these data? Right now, we're so early in the process, in my opinion, that the individuals who are putting together these analytical processes are the manufacturers or the owners of the software companies, and they're saying, do it this way, do it that way. I think, unfortunately, we don't have a publication about which I have an awareness for this at this point. And it looks like our other expert is joining, Larry Sklar. Hi, Larry.
Hi.
Hi, Larry. Thank you for that answer, Nathan. And hi, Larry. Thank you so much for joining us. If I can put you on the spot straight away, Larry, I do have a question here for you. The question is, do you see any comparison between high throughput flow cytometry and the Amazon delivery model?
Yes, thank you. When we thought about using flow cytometry for drug discovery, there was a question about what is the business model? And so the idea is that people sell flow cytometers, people sell reagents for flow cytometers, but in the Amazon delivery model, you actually get paid for delivering samples. And so we thought that high throughput flow cytometry would actually be a business model in which there would be value added by being able to deliver samples in a particular way, in a particular high throughput way. And that was the correspondence to the idea that this is like an Amazon business model, that you don't necessarily produce new samples or new machines, but you get paid for delivering samples at high throughput.
Absolutely. Thank you for that. I have another question here for you, Nathan, which is, during the panel discussion that you moderated earlier, you posed the question, what do you expect in this field over the next 5 years? Can I ask what your answer is to that question?
Oh, I guess I shouldn't ask questions, the answers to which I don't, I'm unprepared to give, but I know I am prepared. I am in the clinical realm, obviously, from my talk presented earlier today. I'm looking at really pharmacodynamic biomarkers as well as predictive biomarkers. Really the focus of this symposium is flow cytometry. And for many of us who specialize in flow cytometry, we have tunnel vision. My vision for the next 5 years is that flow cytometry is integrated with other sources, or flow cytometry data are more fully integrated with other sources of data. And we're beginning to see some of this from you know, the area of imaging cytometry. This is the DVS instrument where, you know, they take slides and they blast off, you know, the top layer of cells and suck it into a tube and do time of flight mass spec, and then use algorithms to recapitulate the image. And many people are aware of this technology. I think that that's where I would like it to go. We take, you know, and more fully integrate these data sets. Because for me, as someone in drug development, I want to know as much as possible about the compound that we're putting into animals or humans. And I get a little picture of it with flow cytometry. I want to get more of a picture by using other technologies, or more specifically integrating either the flow technology or the flow data into larger data sets. So that's where I think we're going.
Thank you so much for that. Larry, another question for you. Where does drug discovery fit into the portfolio of federally funded research?
So in the United States, there have been a number of initiatives that have followed up on the human genome. And after the Human Genome Project, there was then a project called the Molecular Libraries and Imaging Program, where there was a competition to see which tools would wind up being the most successful. And it was then that flow cytometry entered the arena of discovery through the Molecular Libraries Initiative. Initiatives are ongoing at NIH that have to do with looking at what have been thought to be undruggable targets or orphan receptors. And so, for example, NIH now has an initiative called Illuminating the Druggable Genome. And that initiative focuses on kinases, GPCRs, and ion channels. At the same time, in the United States, various institutes at the NIH are interested in supporting drug discovery. So, for example, the National Institute of Allergy and Infectious Disease, which has over time been known largely for their work in vaccines, has now programs associated with pathogens such as bacteria and fungus. So the enterprise of drug discovery And flow cytometry in the US remains active and is espoused or sponsored by many of the disease areas of interest.
That's great. Thank you so much for that answer. I have a question here that perhaps both of you may be able to contribute to. Again, it's in reference to your panel discussion earlier. The question is an analysis question. In results from a flow analyzer with multiple markers to count cell populations, how do you control for batch effect to reliably compare samples? Are control samples repeated across batches, e.g., FX leukopak, or another method?
Nathan, do you want to go first?
Sure, I can speak from the perspective of clinical trials or preclinical, so toxicology studies. So we routinely run, so the question was really about batch effect. We routinely develop quality control specimens when we validate our assays. And so oftentimes this can be, someone, the questioner mentioned a leukopak, but we oftentimes will use other quality control materials. Peripheral blood mononuclear cells that are cryopreserved, of course, is one, but there are companies that generate control materials that are preserved whole cells. And there are even newer companies, in particular in the Bay Area in California, where there's a lot of flow cytometry going on, a lot of manufacturers and software companies who make flow cytometry software. There are new companies popping up that generate synthetic cells. So these are fascinating, and they can even load these with nuclear material, so they will stain and for nuclear dyes. But to control for batch effects, yes, we typically use a preserved cell type that's run on each plate or with each day of the experiment, and we monitor the performance of that QC over the course of the time. Larry, would you like to follow up?
Yeah, so in discovery, there are also ways of running controls. There is a statistical factor called Z-primed. And what you wind up doing typically in high throughput plate analysis is you devote entire columns of your plate to negative and positive controls. So for example, you would run, let's say in a 384 well plate, 324 you test 320 compounds in 20 rows of 16, and then in the other 20 columns of 16, and then in the other columns, you have positive and negative controls. So on each plate, you have the statistics for the performance of the separation between the positive and the negative controls, and then all of the samples. And typically what people say in this statistic that a Z-primed of 0.5 or greater, the plate passes. And so that's done on every plate every day. And you can actually compare the performance of the assay not only as the day goes on, but from day to day. And so you have an absolute measure of whether your assay is performing over time.
I'd like to follow up just briefly, because that's a great example. And while I made of really how to do it, and the follow-up I want to make is that while we don't use a Z-prime, we would set acceptance criteria around the QC control from batch to batch. And I didn't mention how we did that because it can be done in numerous ways, but Larry triggered in my mind. I think in answering these questions, it's not enough to say that we use a reagent or this reagent, but rather that we run the QC multiple times to develop a confidence interval around whatever parameter, if that's number of CD4 positive cells or expression levels of CD4. And we know with a tight degree of confidence what that should look like from plate to plate. And if it tests outside of that, of course, we either rerun the plate or we investigate further. But he brings up a good point. There needs to be some sort of statistical basis for, controlling for this batch effect.
Fantastic. Thank you so much both for that detailed answer. That was great. And unfortunately, I think that's all the questions that we're going to have time for today. So thank you both for your time and for your expertise as well in this session, in your presentations, and in the panel discussion also. For our attendees, if you did have any further questions for our speakers, please do reach out to them via the platform. I'm sure they'd be really delighted to speak with you and to answer your queries. So thank you again both, and that brings the event to a close now. So thank you so much everyone for joining us, for sharing in flow cytometry and everything such as that. And a quick reminder that we do have our on-demand library available. So if you missed any sessions, then please do go to there and catch up on anything that you might have missed. So thank you so much.